Hi, I'm Nicole Tessier, and today we will be looking at a brief overview of implantation and early embryo development. We will be focusing on the events before implantation of the embryo in the uterus, the extra embryonic membranes that form around the developing embryo, the steps of implantation, the main hormones produced during pregnancy, and finally we will briefly touch on the triggers for labor and delivery. So before implantation can occur, the sperm must fertilize the egg, which it does in the distal region of the fallopian tube. In order to fertilize the egg, the sperm must first penetrate the granulosa cells of the egg, as well as the zona pellucida, or the protective coating around the egg. In order to do this, the sperm undergoes an acrosomal reaction. When this happens, enzymes are released from the acrosome in the head of the sperm, which allows the sperm to penetrate that protective outer coating. The fusion of sperm and eggs triggers a cortical reaction, in which cortical granules from the cytoplasm of the egg secrete chemicals that will alter the membrane of the egg, preventing polyspermy, or the fertilization of one egg by multiple sperm. After this occurs, it takes about 4 to 5 days for the embryo to travel the span of the fallopian tube into the uterine cavity. It does this with the help of progesterone, which relaxes the smooth muscles of the fallopian tube. Once in the uterine cavity, the developing embryo is then known as a blastocyst, which is a hollow ball that consists of approximately 100 cells. And there's an image of a blastocyst shown on this slide. You can see here the outer layer of cells, the hollow inner section, as well as the inner cell mass. Now, the extra embryonic membranes are all formed from parts of the blastocyst. The outer layer of cells from the blastocyst form the chorion, which will enclose the embryo and eventually go on to form the placenta. The inner cell mass of the blastocyst will form the amnion, which secretes amniotic fluid, in which the embryo is floating during development. The alanatois, which will become part of the umbilical cord. And finally, the yolk sac, which will degenerate early in human development. Now we will look at the process of implantation of the developing embryo in the uterine wall. So the blastocyst will secrete enzymes that allow it to invade the endometrium layer of the uterus. The endometrial cells will then grow around the blastocyst until it is completely engulfed within the endometrium. The chorionic villi, which are formed from these cells that will eventually become the placenta, will then penetrate into the vascularization of the endometrium. They will secrete enzymes that will break down the walls of the maternal blood vessels, which will result in the villi being surrounded by pools of maternal blood. The maternal blood and the fetal blood do not mix. However, nutrients, gases, and wastes are exchanged. The placenta will continue to grow throughout pregnancy until delivery, when it reaches its maximum size of about 20 centimeters in diameter. During this growth, it will receive up to 10% of the total maternal cardiac output at any given time. Next, we will look at the hormones that are produced during pregnancy. The first one we will look at is human chorionic gonadotropin, or HCG. And this hormone allows the corpus luteum to continue to secrete progesterone, as well as keep the endometrium intact. It also stimulates testosterone production by the developing testes in male fetuses. HCG is also the chemical that is detected by pregnancy tests, such as those shown in the figure on this slide. Now, HCG is structurally related to luteinizing hormone, and as such will bind to luteinizing hormone receptors. The next hormone we will look at is the human chorionic somatomamotropin, or HCS. And HCS is structurally related to growth hormone, and it is believed that it may contribute to lactation. Despite that belief, a genetic defect in which HCS is not produced in the mother will still result in adequate breast development as well as milk production. The main function of HCS is to alter the mother's glucose and fatty acid metabolism in order to support fetal growth in the body. Approximately 4% of women will develop gestational diabetes, which is characterized by an increase in blood glucose levels due to a resistance of insulin, similar to that seen in type 2 diabetes. After delivery, the glucose metabolism usually returns to normal in most mothers. However, the mother and the offspring are now at a greater risk of developing type 2 diabetes later in life. The next hormone produced is estrogen, which will contribute to the development of milk-secreting ducts of the breasts. And finally, progesterone, which is essential for maintaining the endometrium and helps suppress uterine contractions. Now, all of these hormones are secreted by the placenta, but for approximately the first 7 weeks of gestation, progesterone will still be produced by the corpus luteum until it disintegrates and the placenta takes over as the primary progesterone production source. And finally, we will look at the triggers of labor and delivery. Now, these include cervical stretch, which will trigger the uterus to contract, as shown in the figure, and it will also trigger the release of oxytocin from the posterior pituitary. And oxytocin will then in turn trigger uterine contractions as well. Now, it is believed that the fetus itself can signal when development has completed, and this is seen by an increase in corticotropin releasing hormone in the mother right before delivery. And finally, the hormone relaxin, which is used to aid in delivery, will soften the cervix and loosen ligaments holding the pelvic bones together.